Okay, we are live and recording. Maybe we can wait a few seconds for everyone to join. This is a good day. This is a very good day to begin a new year. Amazing talks here. And I will start my intro in 10 seconds. Okay, hello everyone. My name is Ali Mohebi. I'm a postdoc at UCSF, and I am your host today at this 8th episode of Neurolaunchpad. This is a seminar series by and for trainees. You can find us on Twitter at Neurolaunchpad or visit our website for more information. You can also register to receive weekly reminder emails through our websites. That it is my honor and pleasure to introduce our moderators. For today, Dr. Jesse Goldsberry, the amazing Jesse Goldsberry, is a physician scientist and a professor of neurobiology and behavior at Cornell University. He is running a unique and fascinating program investigating motor learning and performance across multiple species, songbirds, rodents, and recently parakeets. His co-moderator today is soon-to-be Dr. Lindsay Cameron. She's a neuroscience student at UC Davis. She also has a fascinating research program studying the therapeutic actions of psychedelic compounds. So without further ado, I will let Jesse to start and introduce the amazing speakers for today. Thanks. Jesse?
Thanks, Ali. Really interesting session. We're going to explore functions of dopamine in settings that are maybe beyond the classic learning and reward prediction error frameworks in natural behaviors. And I'm really excited for this session. It's well matched, these two next talks. First, we have Mor Ben-Tov from Rich Mooney's lab at Duke. She did her PhD at the Ben Gurion University of the Negev with Ronen Segev, where she did really important work on visual search in the archerfish, and keeping her neuroethological roots alive, transitioned to the songbird. And we're going to hear a really interesting story on a dopaminergic projection to a cortical nucleus called HVC, and what this projection may do in the context of courtship. And so without further ado, Mor, please take the mic.
Okay. Oops, sorry. Can you see my slides? I hope the answer is yes. Thank you so much for the organizers for this opportunity to present my work here. And today I'm going to talk about a neural hub that coordinates learned and innate courtship behaviors. So in Rich's lab, we study zebra finches. And the zebra finches are an excellent model, animal model, mostly due to their ability to culturally transmit their song from one generation to the other. So the father, the tutor zebra finch, sings to his son, to his pupil, and throughout development, the pupil memorizes and imitates the song. And then as he reaches adulthood, he can sing a precise copy of his father's song. And for that reason, in many of the songbird talks, you would see a timeline like that describes the process of song learning. But if we zoom out and we look at the timeline of the entire lifespan of a bird, we will see that this song learning period is relatively brief comparing to the entire lifespan of the bird that could reach up to 4 years in the wild. So in most of his life, the male zebra finch needs to perform this stable song, mostly in order to court a female. And again, this is the well-known feature of the zebra finch, a song, and you can see a stereotype sequence of syllables here, A, B, C, D, and E. That's a spectrogram to visualize the auditory output. So the x-axis is time and the y-axis is frequency. But zebra finch has more courtship behaviors, and some of them are innate. So a vocal element, an innate vocal element of his courtship behavior is those innate calls or introductory notes. And these are those three elements that the bird produces just before he starts singing. And those are, again, innate vocalization that even males that were raised in a complete isolation will still produce normal introductory notes. And zebra finches, similar to other vertebrates, have non-vocal courtship behaviors such as orientation towards the potential mate, pursue, some sort of a dance, and of course mating. And here is a video of courtship behavior. That's the male zebra finch, and that's the female. And I hope you would be able to hear it well. So looking at this complete courtship behavior, we wanted to ask two questions. Sorry. One is what circuits underlie sexually motivated singing? And the second one is how are the innate and learned behaviors are being coordinated in the male zebra finch's brain? So that's a very simplified view of the male zebra finch's brain. And I've highlighted here two areas that are part of the song system, which is a specialized circuit for song learning and song production. And today I'm going to focus mainly on HVC, which is a telencephalic nucleus that is very important for both processes. And one candidate that could convey motivational or sexual inputs to HVC is the midbrain dopaminergic group A11, which is the major source of dopamine to HVC. And also from other songbirds, we know that it receives information or inputs from preoptic areas, an area that is well known to be involved in sexual behaviors and arousal. So in order to have a control way to study courtship behavior, we set up this following paradigm. So we put a male zebra finch and a female zebra finch in the same cage, and we put in the middle an electronic glass that was opaque for most of the time. And zebra finches mainly rely on visual input. So when they can't see each other, the male zebra finch won't sing to the female. And then by clicking a button from outside of the cage, we can make this glass transparent, and then the birds are visible to each other. Then we can study the male's behavior without any interruptions from the female. Because many times when the male tries to court the female, she escapes, but now they have this boundary in between them, so we can study the male in a very controlled way. So here's an example of this behavior in the setup. So that's the male and that's the female, and the glass is over here. And when the frame is red, the male cannot see the female. When it turns green, the female is visible to him. I hope you could hear it. So I hope you could appreciate it. As soon as the male saw the female, he immediately started singing and starting to approach towards the female, and he did this big swipes. These are all parts of his courtship behavior. Okay, so to start study this circuit, we injected 6-hydroxydopamine to HVC to abolish the dopaminergic projections in HVC. And we waited a few days, and here is a video showing the male's behavior after this lesion. So in case you didn't hear, the male produced lots of vocalizations, but none of them was a song. But it still did jump towards the female, moved around, look at her, but didn't produce any song. And if we measure the singing rate before and after this lesion, we can see that even almost 10 days after the lesion, the males couldn't almost produce any of those female-directed songs. But as you could see in the videos, the other aspects of courtship behavior remained intact, and I will talk about it later in the talk. Then we went one synapse back, and we wanted to see what's the role of A11 in this behavior. So we did a similar lesion. We injected 6-hydroxydopamine bilaterally to A11. And again, this is a video taken a few days after the lesion. So this time I didn't have any audio issues because the bird didn't produce any type of vocalization, not the learned aspect and not the innate ones. And again, if we look at singing rates, this group of birds with 6-hydroxy lesion, 6-hydroxydopamine lesion in A11, again, didn't produce almost any female-directed song, even 10 days after the lesion. So then one possibility is that we just abolish their motor ability to sing. So luckily for us, zebra finches sing in two distinct social contexts. So they sing to the female, that's a female-directed singing, but they also sing in complete isolation. It's like a practice mode for them. And if you look at the two spectrograms, you couldn't see very obvious differences between these two types of song, but they do have subtle differences, which are very important for the females. Because if we'll put two speakers, one presenting the male undirected songs and the other one playing the male directed songs, the female will go towards the speaker that present the male's directed songs. So the male need to know how to modify their song in order to attract the females. And if we look at the singing rates in both the directed and undirected context, we can see that although they couldn't sing directed songs, birds in both experimental groups could recover to some extent their ability to sing undirected songs, meaning that it's not just a motor deficit, it's really singing in one specific social context when they sing to the female. Next, we wanted to explore the other elements of courtship behavior, the innate behavior. So we started with the vocal courtship behaviors, the introductory notes. So again, that's a reminder to a normal controlled zebra finch song. So he performs a few introductory notes and then a long bow that is composed of several motifs. And if we look at a bird that had a 6-hydroxydopamine lesion in HVC, we can see many instances in which they just produced long strings of introductory notes without producing the learned song. And on the other hand, if we look at one example of a bird with 6-hydroxydopamine lesion in A11, we can see that they barely produce any type of vocalization, not the innate part and not the learned one. And those two elements are the female calls trying to maybe attract the male. And we can see it again in the group data that the birds with the dopamine lesion in HVC produced many more introductory notes than the group with 6-hydroxydopamine in A11. So next, we wanted to explore the non-vocal elements of courtship behavior. And in order to do that, we used Deep Lab Cut. I'm sure many of you know this tool. You can mark body parts on the zebra finch's body, and then for each frame, you get a skeleton of the zebra finch. And from those data, you can calculate the bird's position in every time point, and also the bird's head orientation relative to the female position in every frame. So first, we started with the bird's position. So this is a heat map. This is the cage, and the female, this is the male side of the cage, and the female is on the right side. And you can see here that as soon as we make the screen transparent, and the male zebra finch sees the female, they immediately jump towards the female side of the screen and hop around this edge. That's why you can see this red stripe over here. When we looked at the group of birds with 6-hydroxydopamine lesion in HVC, you can see a similar behavior. You can see a very dark red stripe in this side of the cage, meaning that they still jumped towards the female and hopped around to be close to her. On the other hand, when we look at the group with the 6-hydroxydopamine in A11, you can see a bit of a different behavior. They do have some tendency to move towards the screen, but they also like to just go back to the middle of the cage and sit on their food dish. And if we look at the group data, again, we can see that the HVC group moves towards the female while the A11 lesion group moves far away from the female. And this cannot be accounted for an overall movement. Again, we didn't abolish any of their motor skills because both groups move roughly the same amount of distance while the female was present. Then last, we calculated the
self-initiated movements like picking up a cup, yet they can react to sensory cues with relative ease. If you take a football and you throw it to a person with Parkinson's disease, she'll be able to move nearly as well as a healthy person to catch or avoid that ball. This so-called paradoxical kinesia is so striking that assistive technologies exploit it. For example, there are walkers that display visual cues on the ground that prompt patients when to take steps. This suggests that whatever is going wrong in Parkinson's disease may be the critical clue in helping us figure out what the brain does to self-initiate movement. Now, we've known for decades that loss of dopamine neurons from the substantia nigra causes Parkinson's, which suggests that dopamine neurons could be vital to this self-initiating process. But despite this, precisely how degeneration gives rise to the symptoms of Parkinson's remains really poorly understood, and it's actually been surprisingly difficult to ascribe a role of the endogenous dopaminergic system to movement initiation. In fact, the Dudman lab recently showed that normal levels of dopaminergic firing fail to elicit movement initiation, which suggests that whatever dopamine neurons are doing in generating self-initiated movement has to be more subtle than directly producing it. But there's one other important observation from Parkinson's that might give us a clue, and that's this mysterious slowing down that patients experience, both of movement, but more generally of cognition, exhibited by phenomena like perseveration, which came up in the discussion we just had, which can be thought of as, as I put it before, the inability to nimbly and appropriately transition between behavioral states. And in fact, cognitive slowing in Parkinson's can be objectively measured with self-timed movement tasks, in which a subject receives a cue at time 0 and then has to decide when to move to report when they think a target interval has passed. So here we might expect the subject to move around this time. Now, interestingly, Parkinson's patients tend to move late in these tasks, as though their internal clock were somehow slowed down. Conversely, if we add dopamine to the brain with drugs like amphetamines, we tend to speed up this putative internal clock, resulting in early self-time movement. So because of this apparent connection between dopamine, movement initiation, and timing, we suspected that rather than directly producing movement, dopamine neurons might instead control the timing of when some intended movement is going to occur. So returning to this question of how the motor cortex gets revved up into its coordinated activity to trigger movement, it's intriguing to note that dopamine neurons that are lost in Parkinson's project densely to the dorsal striatum, where they exert a push-pull effect on medium spiny neurons of the direct and indirect pathways. Now, these pathways form parallel positive and negative feedback loops to the cortex. And in an electronics analogy, you might imagine that these pathways could form a sort of cortical amplifier, with the direct pathway tending to enhance cortical activity, and the indirect pathway may be tending to keep this activity in check. In this view, the motor cortex might be able to self-initiate coordinated activity when the gain of the amplifier is just right, or put differently, when the balance of the direct and indirect pathways is tipped in favor of the direct pathway. In this case, dopaminergic activity might influence the timing of movement onset by tuning this balance, tending to push it toward the direct pathway, and thereby pushing the system toward the moment of movement onset. So with this hypothesis in mind, we set out in this project to address two questions. First, does the activity of dopamine neurons explain the timing of self-initiated movement? That is to say, if we observe these neurons as animals prepare to move, will this activity inform us of when that movement will occur? And second, can dopaminergic activity control the timing of movement onset? So in other words, if we causally alter the activity of dopamine neurons using optogenetics, can we influence when a movement actually happens? To address these questions, we recorded and manipulated dopaminergic activity in mice trained to make self-initiated movements. But to study self-initiated movement meaningfully, we had to create a situation in which the mouse had to rely on its own internal processes rather than some overt cue to decide when to move. Because of course we want to distinguish these special self-initiated movements from reactions which don't seem to be so dependent on basal ganglia machinery. To fulfill these criteria, we trained mice to do a self-timed movement task like I described in a couple slides ago, in which the mouse receives a cue and then has to wait for at least 3 seconds before licking to trigger reward, in the absence of any sensory cues as to when it should actually do this. Now, because the mouse has to rely on its internal process of self-timing to decide when to move, these movements implicitly fall under this umbrella of what we operationally would consider a self-initiated movement. But there's actually something very handy about self-timed movement, and that's that mice, like humans, are pretty good at timing, but their time behavior is highly variable. This is well known across the timing literature. When you train a mouse to lick after a 3-second delay, the result is a wide distribution of response times spanning more than 7 seconds. And here I'm just plotting the first lick the animal makes because we only consider that first lick that the animal does to be self-timed. Now, it's important to note that this behavioral variability isn't a bug in this task. It's actually a critical feature. In fact, it's exactly what we need to probe the question of what the brain is doing to self-initiate its own movements, because with it, we have a handle to ask this question. What is the brain doing differently on the trials where the animal decides to move relatively early versus these trials where the animal decides to move relatively late? Our hypothesis is that animals' propensity to move depends on the balance between direct and indirect pathways, which may be tuned by dopamine. So we would expect if this balance favors movement, this would cause the mouse to move earlier on a given trial, and thus dopamine may be relatively high on these trials. But conversely, if the animal moves later, we might expect this balance was tipped away from movement, and thus dopamine may have been relatively low. So to test this hypothesis, we observed the activity of dopamine neurons during the timing task and related their trial-by-trial activity to the relative timing of the mouse's movements. To do so, we took a fiber photometry approach to record the activity of dopamine neurons in the substantia nigra, which remember is the population that gets lost in Parkinson's. We specifically expressed the fluorescent calcium indicator GCAMP6F in these dopamine neurons using DAC-Cre mice with a Cre-dependent virus. This protein diffuses throughout the cell and axon, allowing us to record population activity both at the cell bodies and at the axon terminals in the striatum, where these cells are poised to interact with the direct and indirect pathways. Now, we additionally expressed an inert fluorophore called TD tomato, and the fluorescence of this fluorophore does not modulate in response to neural firing. Instead, the modulation in this channel is used to capture optical artifacts that could be caused by the recording site moving relative to the optic fiber when the animal might be moving, vibrations that could happen in the rig, which we can then use to assess the reliability of the GCAMP signal and correct it for optical artifacts. Now, keep in mind that fiber photometry is not the imaging of single neurons. It's instead an average fluorescence from a population of neurons. Now, the mice completing the self-timed task are water deprived and they're head fixed, and they complete hundreds of trials of this timing task per session. On each trial, they receive a brief LED and tone cue to start timing, after which the mouse has to decide when to lick in the absence of any further cues. The mouse's goal should always be to make its first lick during the reward window between 3.3 to 7 seconds after the cue, because if its first lick is too early or too late, it won't receive any juice for the reward, or any juice reward for the trial. Now, importantly, nothing the mouse does will make the trial time course go by any faster, so there's no advantage to the mouse to licking early. Importantly, the recorded activity of dopamine neurons can be influenced by what I'll call nuisance movements, and that is some kind of body movement other than licking that could be happening during the trial period. So we have to account for these when analyzing the neural signals and attempting to relate this activity to the process of self-timing. So thus, while the mice do the task, we track their behavior using several movement sensors, including the TD tomato control channel that we mentioned before, a neck EMG that detects muscle activity related to licking, a back-mounted accelerometer to detect body movements, and high-speed video camera for everything else. Now, I first want to show you the simplest possible picture of what we found when recording from dopaminergic cell bodies. So this is an average of GCAMP signals across all trials from one mouse, where the response fell between 0.7 to 3.3 seconds. Those are the unrewarded trials for making, this response over here consistent. On the x-axis is time here, but there are actually two parts to the plot. So here's what's going on. On the left-hand side is a cue triggered average because we want to show you the signals that are related to the cue. But because the lick is happening at different times relative to the cue, we needed to split this plot here on the x-axis at this axis break so that we could also align the responses to the movement time. And that's what's being shown here on the right. Now, the signals here are unfolding over the course of seconds. Let's focus on the clear things that we can see right away. Well, first we notice there's a clear cue-related response, and there's also this lick-related spike. And neither of these was too surprising since dopamine neurons are known to fire transiently in response to salient cues and at the onset of movement. But what was surprising was this third thing we saw, namely an apparent ramp between the time of the cue and the time of the movement. This signal starts several seconds before the movement occurs, and it appears as though dopaminergic activity is ramping up to the time of the movement. Now, the rising of this ramp made us think of a timer, so we asked whether this ramp might be related to the mouse's timing. To test this idea, we pooled the GCamp responses by the time of the movement relative to the cue. So first, I'm just showing you the trials where the first lick occurred between 4 to 4.25 seconds. And we see the same rough patterns as before. Now, if we added another movement time, such as this pooled trial bin, where the movements occur between 1 to 1.25 seconds, we start to notice some differences in the dopaminergic signaling. First, we notice that there seems to be a subtle difference in the baseline activity before the cue even occurs. And this difference seems to become more pronounced as the trial interval elapses. In fact, after the cue, we never really dip down as far as we did for the late trials in the early trial pool. And instead, we ramp up much more quickly. Nevertheless, despite these differences, it seems that we ramp to about the same amplitude of dopaminergic signaling, regardless of when the animal moved. If we fill in some pooled trials in between, we see that this pattern is preserved. Once again, the baselines start from a similar point, but they start to diverge even a little bit before the cue happens, after which follows a clear fanning of responses that's already predictive by eye of when movement will occur, starting more than 4 seconds before the movement onset, which is a long time for neurons. I mean, so think about it. Just by looking at the dopaminergic signal, we can learn something about what the mouse is going to do up to seconds in the future. I thought that was pretty wild. But we didn't just see this pattern in one animal, we actually saw it in every animal we tested. When we averaged together signals from all 12 mice, we found this pattern becomes strikingly clear. And to more clearly examine when examine the pre-movement responses in this figure, I've truncated the average traces at the time of the movement onset, so we don't have any distracting stuff going on in the background. Now, by eye, we can see the dopaminergic signal is highly predictive of movement timing, starting even before the cue occurs. And it starts to fan out by the time of the cue, and then starts to diverge. And we see this ramping dynamic occurring over the timing interval. In contrast, TD tomato control data doesn't show any of these patterns. And I'll just note briefly that we found similar dynamics in GCamp signals at dopaminergic axon terminals in the dorsal striatum, as well as in striatal dopamine release recorded with the novel dopamine indicators D-Light and DA2M, suggesting that these dopaminergic signals are in fact passed downstream to the direct and indirect pathways, which are positioned to tune the timing of movement onset. Now, because these signals look reminiscent of a timer, we asked whether a simple threshold model for GCamp might predict movement timing. And our eyes tell us, looking at these averages, of course, this works great on the grand average data set. And here I'm showing just one arbitrary threshold, but you can draw this horizontal threshold at any point along the y-axis here, and you would be able to do a pretty good job of predicting the movement time. And although I'm showing the results here for the averages, dopaminergic dynamics were highly predictive of movement timing on single trials, independent of task variables. Now, one caveat of the view I've just shown you is that dopaminergic neurons are active around the time of movement, and photometry is also prone to optical and mechanical artifacts. And because of this, we needed to be sure that neither the baseline nor ramping differences on trials of different length were occurring due to some difference in behavior of the mice on early versus late trials. It's a little strange to imagine what kind of movements would cause this pattern of activity, but we might imagine that this head-fixed animal might start squirming faster and faster as the time of movement approaches, and maybe that somehow gives you a consistent ramping across all the 12 animals. So we looked at average traces of the movement control channels aligned to the movement onset here at time 0. And what we noticed is that the animals are pretty quiescent during the timing interval. We don't really see very much activity at all in the movement control channel before the movement occurs. And importantly, we don't notice any consistent differences in the movement control channel, whether those be early trials as in blue or these late trials in red. However, we're not missing movements because once the movement does occur, we do see clear and distinctly different patterns of movement activity once the animal has actually licked. So you can actually see the little lick, 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 lick pattern that the animals make when they're rewarded. We found a similar picture in all the movement control channels, making it unlikely that movement or some hidden behavior other than timing could explain the baseline differences or ramps. Nevertheless, we wanted to quantify the contribution of these nuisance movements and task variables to the dopaminergic signal. And as such, we used a GLM encoding model to isolate and quantify the contributions of things unrelated to self-timing, such as optical artifacts, behavioral events like the cue and first lick, and nuisance movements as detected by EMG and/or accelerometer, with the goal of distinguishing contributions from these timing-independent factors from contributions arising from more occult cognitive processes predictive of movement timing, which we represented here as a baseline offset whose amplitude is proportional to the movement time, as well as a temporal stretch feature in which we represented percentages of elapsed time in the self-time interval. And for those in the audience who might be less experienced with GLMs, just keep in mind the goal of what we're doing now is trying to predict the dopaminergic signals. And in so doing, we aim to determine if the ramping and baseline offsets we observed in the averages can be explained by things like nuisance movements, or do we actually need to invoke something more hidden and cognitive like timing? We applied the model to both the GCAMP signals as well as simultaneously recorded TD tomato signals. And we used the TD tomato control signals as a way to assess if the model was actually able to detect nuisance movements and encode them properly. We don't want to just miss them, right? So we used a nested modeling approach in which we applied these nuisance movements and task-related variables first, which allowed the GCAMP model to capture the cue and lick-related transients observed in the averages. However, nuisance movements did not produce the ramps or baseline offsets we observed. Conversely, the timing-independent model captures up to 80% of the variance in the TD tomato signal, showing that the model does indeed capture variance related to nuisance movements and artifacts. When we then nest on top of this the timing-dependent baseline offset and stretched representation of the timing interval, we can generate the full timing-dependent model, which we see robustly improves the GCAMP signal model, leading to a relative median loss improvement of about 27% across the 68 sessions indicated in the dots on the right. Now, importantly, the stretch time feature does not impose any kind of ramping dynamic. So, and we can actually see this in the TD tomato fit because it doesn't find ramping when ramps were not present, nor does the artifact channel further improve by adding the timing dependent features. And I'll just note, we found similar encoding patterns in the SNC axon terminals, as well as extracellular dopamine signals in the striatum. Now, given that dopaminergic dynamics are predictive of movement timing and were not explained by ongoing movements or artifacts, we asked whether causally manipulating the activity of dopamine neurons on randomly interleaved trials would influence movement timing.
Okay, so 2 minutes.
To address this, we optogenetically activated or inhibited dopamine neurons on 30% of trials from the time of the cue up to the time of the first lick. Because dopaminergic activity tended to have a higher baseline and ramp up more rapidly preceding early movements, we expected activation might result in more early movements on stimulated trials compared to unstimulated trials. Conversely, we expected inhibition would result in later self-time movements. To test this idea, we compared the distributions of movement times on stimulated versus unstimulated trials. And here I'm showing these distributions as CDFs, which you can think of as integrals of the behavioral distribution. We found that dopaminergic manipulation bidirectionally tunes the movement distribution, with activation causing early shifts and the inhibition causing late shifting of the distribution, whereas no opsin control animals showed no consistent effect of stimulation on behavior. Importantly, changing the dopaminergic activity did not instantly trigger movement, nor did it prevent movement, suggesting that dopaminergic activity influences timing by controlling some moment-to-moment probability of movement being able to occur. Taken together, these results argue that dopaminergic input to the dorsal striatum predicts and causally influences the timing of movement initiation. So what does this mean in the context of the basal ganglia? Well, the dynamic signals we observe do look reminiscent of something like a ramp to threshold process, wherein the rate of ramping depends on the baseline dopaminergic activity. Now, we're reluctant to say threshold because how a systems level threshold process actually works is pretty speculative, but we might imagine this could be the point at which the direct and indirect pathways critically diverge in their activity, allowing movement to initiate. And while we can't make mechanistic arguments from this data set, it may be possible that Parkinson's patients live in a state much like the mouse when it moves relatively late on a given trial, except more extreme, in which the baseline level of dopaminergic drive is so low that even when patients make a big effort, the system isn't able to ramp up very easily, requiring more time for this process and sometimes failing to reach whatever this threshold might be altogether, resulting in delays in movement and behavioral transitions. To test this idea, more work is needed to understand the dopaminergic activity and how it's related to and processed by downstream elements of the basal ganglia. And in particular, how do cell types within this circuit work together, and how do their interactions go on to causally influence movement initiation? We're excited about the possibility of employing multimodal approaches, such as combining fibrophotometry, optogenetics, and electrode recordings, to be able to simultaneously record and manipulate multiple circuit nodes during self-timing, in an effort to finally get to the bottom of what's really going on in the basal ganglia that makes us move. With that, I want to say thank you especially to my incredible mentor, John Assad, and also to two really great students who did some control experiments, Giulia Spedicato from Italy and Mary Hong, who was at the Chinese Academy of Sciences. And I want to thank all of you for joining us today for Neuro Launchpad, and I definitely want to have some great discussions, so let's get to it.
Great job, Allison. That was very clear and crisp. Really good talk. I'll start with a question, maybe a little background context again, is that, people have been looking at dopaminergic signals associated with upcoming movements since Romo and Schultz, I guess 40 years ago by now. And, more recently, since photometry, the COSTA lab has seen some signals that are preceding upcoming movements. And it seems like what's unique here is this notion of reflecting the ongoing process of timing an upcoming movement, right?
Yes.
So the nice thing about that is it makes these strong predictions and it looks like you're testing, but it seems like maybe the data to support that or not are already out there with respect to the interpretation dependent on the direct and indirect pathways. So in a task like this in either a monkey or a rat, has there been recordings of either GP or SNR that would support or not support the idea that there is this underlying ramp process, like a ramp to threshold in the basal ganglia that would be reading out the way you're interpreting this signal?
Yes, absolutely. Okay, this is a fantastic question. So first of all, we actually kind of had some inkling that this might be the case back from really early work from John's lab in 2003 with Irwin Lee, who was a grad student at the time, where they recorded, with monkeys, you can't tell if it's direct or indirect pathway neurons, but they recorded striatal neurons in monkeys that were preparing to self-time movements. And they did find, indeed, that there was a distinction between movements that were made in reaction to a cue versus movements that were self-timed. And that's that there really wasn't a lot of activity in those pathways until right before the movement when the animal was reacting to a cue. But when the animal had to self-time the response, there was this little rumbling of activity that increased steadily as the time of movement approached, suggesting that maybe there was something going on in that system that was revving up. Now, I can say more recently, my lab mate, Ling Feng Ho, has been working on SNR, doing a similar task to what I've been doing with mice, which, with the mouse tools, of course, we can be a bit more nuanced about how we're recording neurons. So he's been recording SNR neurons and doing a bit of circuit tracing. And what he's found is, very intriguingly, there are a population of neurons in SNR that are ramping down slowly as the time of movement approaches. Now, of course, there's a lot of sign changes, right? SNR is a negative output from basal ganglia. It's tamping down thalamic activity, but it's consistent with what we observe in the sense that perhaps as direct pathway activity is increasing over the course of this interval, this could be shutting down units within the SNR that could then be leading up to the release of the moment of movement initiation. So we are working towards that slowly. We're trying to move in, I think, from both sides to try to probe all the different spots in the circuit to get at that. But yeah, there does seem to be intriguing evidence, and specifically related to self-timing. And I think that's a critical thing that wasn't seen in, for example, the classic studies with direct and indirect pathway, which were really examining reactive movements. They weren't really looking at these movements that depended on the animal making its own decisions of when to go. This seems to be a really important feature if you're going to study these movements, is that the animal has to be allowed to do what it wants, as opposed to make a short latency reaction, within like a few hundred milliseconds to a cue. I hope that answers that question.
Yeah. Thanks. Lindsay?
Hi, great talk. Okay, so I will jump in with the first audience question. How does the role of timing fit together with the role of dopamine and reinforcement?
Yeah, such a good question. All right, I'm gonna just point on my slide. We have a little theory paper that I put a little QR code for. So It's an interesting question. Now, I'm at Harvard, so I tend to be influenced by the Harvard School of Thought on this, and our colleagues in now Cheetah's lab, including Sam Gershman, who recently became a professor, they've been looking into how reinforcement learning can be made consistent, or at least can be made sense of in terms of dopaminergic ramps. You know, we're not the first group to report dopaminergic ramps. They've been seen in contexts like, you know, goal-motivated navigation, as well as these multi-step bandit tasks. So Sam Gershman's lab developed a model with a very talented classmate of mine as well, John Michael, who were investigating the possibility of whether or not value or RPE-like sample signals could give rise to ramping in the context of these behaviors. So I'll just say in our task, our results are super consistent with both a value model and an RPE model. You know, you can't distinguish that from our results, because basically the value function of our task is essentially you have a value landscape that's just going up, because as the time of movement approaches, value is increasing up until that moment, right? So they look essentially identical. Now, one nice thing about an RPE-based model is that it's, you know, under conditions of state uncertainty, RPE is pretty much proportional to the derivative of the value function. That gives you a little Q-related response, and you can model the responses in our task very nicely with RPE-like signals using the Gershman and Michael model. However, one nice and attractive feature of the RPE model, and I'm not going to put my hat on anything, because I'm sure there's a lot more complicated things going on in the system besides just RPE or value, but an intriguing possibility is that we were able to explain both the signals in our task as well as dynamics in another perceptual timing task using this RPE-like model. And this work is, you know, pretty conceptual at this point, but we're definitely interested in pursuing that a little bit further. As far as I'm concerned, I'm agnostic to that argument. It doesn't really matter to me one way or the other, but I do think it's really interesting and nice that you could potentially parsimoniously explain the activity of this system across many different domains of cognition and across different timing tasks using a temporal difference learning model. So I would definitely encourage people to check out that paper if you're interested in it, and definitely give me feedback, because that one is one that I'm still working on. Mm-hmm.
Awesome. Great answer. Okay, so the next question is, are the dopamine ramps apparent on all trials, or do they only appear after the animal has had experience with the task and has learned the 3-second target time?
I love that question. Okay, so, oh gosh, I prepared slides for almost everything except for the task learning, which now I'm kicking myself about. The way that we trick the animals into doing the timing task is we take advantage of an observation from Pavlovian conditioned learning, which is that if you give animals a Pavlovian reward at a consistent time, the animals will start to anticipate that reward time. And so in our task, they start anticipating a Pavlovian reward at 5 seconds, and that's how we get them to start self-triggering their own movements. And we train them to do that all the time. Now, what we see is I record photometry signals throughout training. So in that first day of training, no, I don't see ramps at all. When I start to see ramps is really in the second day of training. And what we observe is that the signals do start ramping up slowly as the animals start to have experience self-triggering their own rewards. And what's interesting at that point is when we're in that early stage of training, it's like a hybrid task. There's both Pavlovian and self-initiated movements happening. And anecdotally, I can say that the ramps that seem to be evolving on those Pavlovian trials where the animal doesn't self-initiate, it's like they seem to be evolving more slowly, as if the animal were going to self-initiate a lick, but just hadn't quite reached the right point before the Pavlovian reward came along. I hope that's clear. That may not be super clear, but yes, it does evolve with training. I'll say that. And I'll say too, some groups have said that, you know, these signals kind of go away over time. I've trained my animals for a pretty long time. I'd say these signals only get stronger as they go until your fluorophore starts, you know, dying out. But we are limited in the window in which we can look with GCamp. It starts to degrade over time. That helps.
Do I have time for one more question? Is that okay? Okay. So this person asked a question which I had a very similar question, which is, could this be considered a neural correlate to working memory?
Yeah, okay. This reminds me too. So when I was in med school, I started getting interested in neuroscience because of a very similar question. I remember asking John, who was my professor at the time, like, how does the brain like hang on to memories as you're thinking about things? And, you know, of course, it's not really known, but I do think something that's interesting in this case is that you do have this kind of like recurrence happening where you have like a buildup of signal over time. It's like the animal is keeping track of something. Is it exactly working memory? I guess you could think of it that way, in the sense the animal has a sort of readout of its history. But I would also suggest it isn't necessarily, and we don't have enough evidence to really say that, because I think if it is a working memory sort of thing, I would think that you would need to have some sort of history dependence to this signal. Like if the signal, jumps around a lot, is the animal necessarily going to shoot off a movement at exactly that time, or does the system depend on more of a history of that signal being around and persistent for a while? And I think there's a lot of reasons to think it would be. I mean, for example, dopamine is released into the striatum and it sticks around there on D2 receptors for quite a while. So I would just say I'm going to punt on that one, only because I'm in the process of doing a bit of Markov process versus, history dependent analyses of these signals. And I think I'll have a better answer for you once I've had a chance to look at those more carefully. But it is a really intriguing idea. It's something we're interested in pursuing.
Very cool. Thank you so much.
Thank you.
Okay, thanks. That was awesome. Ali?
So I think unless you have questions, Jesse, I mean, we can wrap it up. We are like 2 minutes after time. And yeah, you go ahead, wrap up.
I mean, I could make one brief question about it. I guess I'll fill the time with this question. It was on my mind. I was going to follow up with you later, but I can ask it now. In your talk, you twice invoked the idea that this signal is ultimately being read out by the direct and indirect pathways through a thalamocortical loop. You invoked the notion of cortex initiating action, and then you invoke the idea that these output signals are going into the thalamus. But I would question that given that decorticate mice can do basically anything, they might be able to do this task. And the recent paper from Sabatini's lab shows that the VLS nigral collicular projection, along with Henry In's lab, which suggests to me that the initiation of lick is going straight out of the basal ganglia right down into the colliculus to initiate the lick. So I would just question the interpretation that you're even involving the thalamocortical loop, but that's a testable idea, so it's not unreasonable, but you might want to consider the alternative too.
That's a fantastic point. And it's interesting that you bring that up, because my current lab mate, Eden Syed, is actually studying superior colliculus and trying to figure out where licking motion comes from. I completely agree. For this particular activity, it could be that the readout is going directly to colliculus. That's kind of the problem with this sort of brainstem-driven movement. And it's true that you might be able to do a little bit better or find something different if you looked at, say, like an arm movement or something. One thing that Ling Feng, who's studying the SNR, is doing in the lab right now is he's actually testing mice on both a licking task and on an arm movement task. So far, he hasn't found a difference in the SNR signals that he observes between the two different modalities. But I definitely agree with you. It is something that remains open-ended. And I think something that's true about this project, which I originally intended on probing all the different areas, and then 6 years passed and, you couldn't quite get there. But right now we're still looking at just this one area, dopamine. We definitely need to explore these other areas. But that's my pie in the sky dream, is to probe everywhere and listen basically to the whole circuit talking. Mm-hmm.
Oh, thanks.
Thank you.
Okay, so with that, let me thank everyone. The moderators and the panelists. Very, very interesting set of talks. Thank you, everyone, and thanks to the attendees. We will see you next week. Please sign up to get our weekly email reminders. And then I'll see the panelists on the other side in a few minutes. All right, thank you, everyone.
Thank you.